All right, now that we have established how microbes are able to cause disease, now we're going to look at diagnosing those infections. All right, so there are three different categories for determining these. So there are phenotypic methods, immunological, and genotypic, and we'll go through each of those in just a minute. But first of all, these can increase point of care diagnosis. So for example, the COVID tests, they can actually be performed by either the patient themselves or at their bedside or where the returning results occur within an hour. So very fast results, they don't have to be sent out, and that just allows the patient care to occur without delay. So it's a very quick method. So our summary table here highlights these three categories. So again, the phenotypic, these are going to be observations using a microscope or a macroscopic, looking at their shape, like morphology, their physiology, susceptibility to different antibiotics or antimicrobials, and their biochemical properties. So looking at their physical characteristics. And we can also use immunological tests where this analyzes microbes using antibodies. So it's not our immune response, but there are created antibodies that detect or bind to different microbes to indicate their presence, or looking at patient antibodies using the antigens. So either using antibodies to detect the microbe or identifying antibodies that are produced by the patient. And then finally, genotypic, this is analyzing the DNA or RNA of the microbe. So first of all, the sites where we can collect this, there are multiple sites depending on which test is being run. But essentially, we have saliva, sputum, and swab that come from the oral route. We can look at blood tests, urine from a clean catch, just meaning trying not to get any resident microbiota directly from the catheters. So skin using a scalpel. We can do vaginal swabs from feces. We can also do spinal taps to look for neurological issues, and we can swab the skin. So lots of variations, again, depending on the test or the potential disease or the causative agent that's expected would determine where we're collecting from. So first of all, regardless of where that is happening from, proper collection is so important. So number one, prompt transportation to the lab or storage. Usually refrigeration is so important, because if you would take say a urine sample and you would just leave it out for a few hours, well, those microbes, whatever is in there, if there is an infection, they're going to keep multiplying. And so that's going to provide an unusual response or an inaccurate response. It would look like the infection was much larger than it actually is. So the transportation and the storage is really important for those. Also, there are special swabs and transport systems that are designed to help maintain stable conditions for many hours, just meaning that the number of microbes that were present as soon as it was collected are the same as whenever it's analyzed. So there's no false or inaccuracies in the results. Now, the clinical form that's used on this has important aspects that we're looking at. So it's very important that the health professionals know how to properly collect, transport, and store those. They label and identify specimens. That's so important, so that there's no mixing of data, and that helps. Also, is important to providing accurate patient history that can be very vital in determining a diagnosis. So the cultivation of the specimen. So once the sample is collected, usually they try to grow whatever the potential microbe is. So there's specialized media that can reveal to help us identify some of those phenotypic things, like the colony appearance. Is it motile? Does it need oxygen or doesn't need oxygen? So these types of media, we have selective media, and this is used to enrich the specimen, so it helps it to grow. We're trying to facilitate and encourage the growth of only the suspected pathogen, because again, especially if we're swabbing the skin, there are tons of microbes there, but we may just be looking at one particular one we think is the cause of the disease. So we want to facilitate or selectively grow that one. And then there are also differential medias, and this helps identify the organism with the distinctive characteristics or fermentation, like how they're using energy. So it may cause the media to turn pink or yellow under certain conditions if a microbe's present. So it helps us differentiate between one organism compared to others that might be present. Now, dichotomous keys are then used. So this is a nice graphic method. It's kind of like a flow chart. So if we look under the microscope and we see that the microbe is coccus shaped, so they're circle, then we did a gram test. If it's gram positive, we would go this direction. If it's gram negative, we would go here. Then we would look at its like oxygen requirements. So it needs oxygen, it doesn't need oxygen. And then it helps us ultimately, by doing these series of tests, helps us to get down to one or a few different potential candidates. So again, it's just kind of a flow chart for identifying specimen. Now the biochemical testing, these are physiological reactions to the bacteria, so with nutrients and substrates to provide evidence of an enzyme system. So do they have certain enzymes present that they could go through and produce a product? So typically these are going to be visualized by a color change. So if we have an unknown microbe and we add a certain substrate for it, if that microbe has the enzyme, then it would produce a product and it would change the color. That would indicate a positive result. If it doesn't have that enzyme, then nothing would happen and it would be a negative result. So again, just another way of identifying, instead of looking at shape, it's looking at some of the enzymes it has. And these can be performed either by machines or manually. So it's cultured in a medium, again, maybe selective, differential, or just something that allows it to grow so that it can produce a certain product. Then the expression of that enzyme is made visible by some type of colored dye. So then no coloration indicates a lack of the enzyme, and then a colored reaction, and it could vary, like how much of the enzyme could cause a greater change or a lesser change. Microbial, antimicrobial susceptibility is also used. So this is just more of an automated system in which the bacteria is exposed to different antimicrobials, so like different antibiotics or something for a particular infection site. So it simultaneously tests for multiple things to see what it's sensitive to and what it isn't. And that can also help identify the pathogen. And then finally, there's actually phage typing. So back to those bacteria infecting viruses, so bacteriophages. So this is where the viruses attack the bacteria in specific ways. So again, if it is the bacteria and you add that bacteriophage, then it would break it down and that would create a reaction. It could help identify, especially things like salmonella. So it's used for tracing the strains in epidemics. So you inoculate, we'll see a picture here in a second, a lawn of the cells. You add the different phages to it, and you look to see for a clearing area to where, again, that bacteriophage is inhibiting the bacteria from growing, indicating that it's sensitive to it. And here's that picture. So again, there's bacteria all over this plate, different phages. Let me get rid of all that. Different bacteriophages would be added to each of these little boxes. But then as we see here in both of these, that bacteriophage was able to inhibit it. So it is sensitive to those two phages. So then determining the significance. So it's important to determine if that isolate is clinically important or it's just a contaminant or part of the normal biota. So is it actually a pathogen or is it just something that happens to be present? So we focus on the number of microbes. So a few E. coli's is normal because it's part of our normal biota, but hundreds of them would indicate there's an active infection. It's growing out of control. Now, the presence of just a single colony of a true pathogen, like Mycobacterium tuberculosis, that would indicate that it's involved in the disease. But if it's part of the normal flora, one of those opportunistic, then again, significant numbers would need to be present. Whereas true pathogens, any of them that are present would indicate it's potentially the cause. Now, some of the drawbacks to these phenotypic methods is it usually requires a bit of time, so up to about a day or so, maybe even longer. So many conditions may be caused from non-culturable organisms, so then if you can't grow them on media, then you can't run the test or look at them under the microscope. So it just leaves an opening that the possibility that the organism is just a simple bystander. So again, you're culturing something of the normal flora, not actually what the pathogen is. So a nice summary table here of these diagnostic phenotypic. So direct examination, microscopy would be an example of that. So that doesn't necessarily involve it and require it to be cultivated or grown. Using those selective or differential media that only identify certain ones or only certain things can grow on them. So that does require, of course, that it has to be grown. Those biochemical tests, again, they have to be grown so that we can look for those different enzymes that are present or absent. The susceptibility requires it to be grown so that we can look at how effective different antimicrobials are at breaking them down. And then some of the miscellaneous, so like especially the phage typing that we talked about, again, it still has to be grown in order for the phages to be added to it. Now, with the immunological methods, this is also the term serology, which is a branch of immunology that deals with diagnostic testing of the serum. So it's based on the concept that antibodies are extremely specific to an antigen. So visualizing interactions between an antibody can be, so adding an antibody that detects a salmonella, it can detect it, it can identify it, or we can just measure antibodies in the person's blood. So we can, say remove the blood, add an antibody that we know binds to salmonella, and then if it's able to bind, we know there's salmonella present, or just measuring the amount if the patient has any antibodies specific to salmonella would indicate they've been exposed to it. So an unknown antibody can be detected using known antigens. So again, we can either look for using the antibody to detect it or looking for the presence of a specific antibody. So the principles of this, so does the patient make antibody toward the microbe, or what is the identity of the microbe? So either we're asking if antibodies are present because they've been exposed to it. So again, we would take the serum out, we would look for antibodies. So we would take a known microbial agent, and if we put them together and there's a reaction, then we know that the patient had to have been making antibodies to that agent. So again, this is looking for the presence of antibodies from the patient. The other place using antibodies for identification of microbes, here we're taking isolated colonies where we don't really know what's growing, and then we add antibodies that are specific to one particular microbe. So in this case, Staphylococcus aureus. So these antibodies only bind to Staph aureus. So if we put them together, we take our colony, we're not sure what it is, and we add this antibody that only binds to Staph aureus. If we get a reaction, then that tells us that our colony over here was Staph aureus. So we can use either method for these serological tests. So some of these diagnostic methods, and there are quite a few. So agglutination, this is where there's clumping whenever the antibody attaches to the cell. So it indicates, again, the presence of antigen. We can get precipitation, and this is where antibodies and antigens bind and it makes more of a solid, so it comes out of solution. Immunochromatography, this is the most common form, the most common form is what's called a lateral flow system, and it's in these prepackaged cartridges, and it's going to create some type of color change whenever there's a reaction that is present. So again, color changing based on the presence of an antibody antigen reaction. There's also a method called a Western blot. This uses electrophoresis, which is an apparatus, a machine. It separates proteins that can either be the antigen or the antibody. And then specific antibodies or antigens are added to look for detection. So we would take a sample, run it through this electrophoresis device that separates out things. We add the antibody or antigen to it to look for a positive reaction. We also have fluorescent antibodies. So there's direct analysis. This is where the unknown specimen is exposed to a known antibody. So again, the antibody would bind if the antigen's present, or we can have an indirect, and this is where the patient's antibody is probed with a fluorescent antibody. So direct is where the fluorescent antibody binds to an antigen. Indirect, it would be detecting the presence of an antibody in the patient's collection. And then an ELISA, this is what's referred to as a sandwiching technique. It uses small little wells to look for antibody and antigen reactions, and then another antibody is used and added on top of it to produce a color change. And then finally, an in vivo test. This is where the antigen is introduced into the patient to cause a reaction. So like the TB skin test. So again, the antigen is put in there, and if the patient has antibodies against tuberculosis, then it produces a reaction that creates kind of a rash in that site. A little bit more about the immunochromatography and that lateral flow test. The COVID test is actually a good example of this, and pregnancy tests are similar, of course, not used to diagnose disease. But what we have here is this kind of plastic cartridge that's on here. There's a place to add our sample, and then the sample, that fluid flows in one direction over the site. And in this case, with the COVID test, antigen is added here. Let me get rid of some of this. So antigen has been, absorbed onto the paper of this cartridge. And so what happens is if the patient is actually producing an antibody to COVID, then it's going to bind to the antigen on the cartridge, and it produces a color change. It produces this banding that occurs. And this is typically, there's a control that would be there too, just to ensure the patient that the test actually worked. So here we have the control. We know it's already been added, so we have an antibody antigen complex that we know will work and react. And then this would be our test. So this would be from the patient that's going over it. So if the patient has the antibody to COVID, it would bind onto this cartridge and produce that color reaction. So this is that immunochromatography. Now, an antibody titer is a concentration of the antibodies that are actually in the sample. So if we're looking for the number of antibodies, that's referred to as a titer. So it's typically serially diluted from the serum into test tubes into smaller and smaller amounts with equal amounts of antigen. And that's defined as the highest dilution that still produces kind of a clumping or that agglutination. So the titers are often used to diagnose autoimmune disorders and also past exposures to certain diseases. Now, the term specificity and sensitivity we've had before, but we're coming back to these two terms. So again, these go back to the false positives and false negatives. So specificity is the property that focuses on a certain antibody or antigen. It doesn't react with unrelated or distantly related antigens. So tests that have high specificity have a low false positive rate. If they're not as specific, though, then they can have high false positives where it looks like there's a reaction, but really it's not because it's not specific enough. Then sensitivity is kind of looking at those false negatives. So this is the detection of even minute quantities. So even if there are only a few antibodies or antigens, it would still produce a positive reaction. So if it's highly sensitive, then there's a low chance of a false negative. So again, if somebody has just kind of started an infection or they don't have very many, if your test, very many antibodies yet, if your test isn't very sensitive, it'll look like a false negative, like they don't have any antibodies. But in fact, your test just wasn't sensitive enough to pick them up. Now, our last category are those genotypic tests. So we have three that we're going to talk about. So we have nucleic acid amplification. Struggled with that one. So the main one here is going to be PCR, polymerase chain reaction. There are a couple different types of this, but it's ultimately increasing the amount of specific DNA sequences. Then also hybridization. So an example is fish fluorescent in situ hybridization, where it's using probes to specifically bind to different sequences to detect the presence of certain DNA. And then there's also whole genome sequencing. This is a high throughput method where it's completely sequencing the entire genome of, say, a microbe to determine what it is, to specifically identify it based on its genetic information. All right, so looking at some of these a little bit more. So again, PCR, polymerase chain reaction, lots of tests use this. This is used in research and lots of other things as well. But its job is to produce multiple copies of DNA or RNA within a short amount of time. It can amplify any even very small, minute quantities to large enough quantities that they can actually undergo diagnostic testing. And it can be performed on a variety of organisms. So bacteria, viruses, protists, or fungi, anything with DNA, it can be, or RNA, can be amplified. So there are a few different types of PCR. Real-time PCR, this uses fluorescent labeling during the amplification process to measure the production. Reverse transcriptase PCR, this involves the creation of DNA from RNA. So again, typically DNA is used to make RNA, but reverse transcriptase is if we have RNA that's present, it can actually be used to go back and make the DNA out of it that then could be tested. A multiplex PCR, this has primers for multiple organisms in a differential diagnosis for the patient's symptoms. And then a pan-bacterial PCR, this is a modification of real-time PCR that uses primers that's common in all bacteria. And then finally, the RT LAMP test as well. Now in hybridization, we're probing for identity. So it's identifying a microbe based on segments or pieces of its genetic material. So the probes are small pieces of DNA or RNA that are known to complement or bind to specific sequences of nucleic acid to a particular microbe. So if they bind, just like the antibody, if it binds to a known antigen, we know that it's present. Similar here, it's just looking at DNA or RNA instead of an antigen or an antibody. So the base pairing, like the A to the T or the C to the G, that probe of the nucleic acid, again, provides evidence that the microbe is present. Now the FISH, the fluorescent in situ hybridization, this, again, fluorescently labels probes to look at intact cells within the patient's specimen or the environmental sample. So glowing cells start occurring to determine the identity of specific microbes. So it's usually used to confirm a diagnosis or identify a component of a biofilm. And so this is just looking at peptide nucleic acid PNA FISH testing for Staph aureus. So here we have a positive blood culture tube. So Staph aureus is present in this blood. A gram stain has been done. It identifies Staphylococcus, again, clusters of circles here. So is it actually Staph aureus though? So then what happens is we get this probe that has a little fluorescent molecule on it. We add that to this sample. If it's Staph aureus, this probe will bind to a specific sequence that only Staph aureus would have. So it gets added and then rinsed. If it attaches, if this is really Staph aureus, then that probe will bind. That fluorescent probe lights up, and you'll see these glowing clusters where that specific DNA or RNA is present. So it indicates this is Staph aureus. So then the whole genome sequencing, now the cost of this has went down. So there was the human microbiome where it completely sequenced the genome of humans, but it took years. It was very expensive. That technology, though, has just went absolutely crazy to where now the cost of that is so low that it's actually commonplace to do whole genome sequencing on microbes in a very short amount of time. So there's rapid analysis, especially for outbreaks and drug-resistant organisms. And there's also this deep sequencing in which a single genome can be scanned and analyzed with a few errors. In case it has undergone like a mutation or something, it would still be detected. Another example is what are referred to as microarrays. So these are plates or chips that have specific gene sequences, and essentially thousands of different infectious agents can be added, and it's selected based on the syndrome being investigated. So again, all the possible players, gene sequences are added, so it can contain, again, bacterial, viral, fungal genes all in one test. So you can just have these chips or these plates with lots of different sequences. You add one sample to it, and it allows the detection of multiple microbes all at one time. So that patient sample where the nucleic acid is isolated and incubated, then the sequencing is labeled on that. So again, if they match, they would hybridize or bind, and then those fluorescent labels, just like with the fish, would light up and be able to identify them. And this is just what this would look like. So again, here we have this plate where we have lots of all these little wells. Each well has a different sequence that would identify and indicate a different microbe. So the sample would be added to it. Again, if there's hybridization to the different sequences, then we have computer analysis. If there is binding, it would combine all the information and identify all the microbes that were present in that sample. And this has been further made into what's referred to as lab on a chip. So it takes advantage to using those small amounts of fluid because it doesn't need a lot to put on that little cartridge. But these chips are designed to diagnose, again, a variety of conditions or tests all at one time. So each little well or each plate could be doing something different. And they employ these different CRISPR techniques. And the biggest impact of these is in developing countries where, again, they don't have access to a lot of technology, or again, storing and transporting is an issue. So it's something that can quickly be done. Again, each well can be diagnosing or detecting something different. So lots of tests are being done on just a small amount of fluid. And then another one here, mass spec. So MALDI-TOF, this uses the technique of mass spec technology to provide rapid and accurate identification within minutes. So again, like our lab on the chip, it's very quick. So it analyzes the protein fingerprint. So instead of looking at DNA or RNA, we're looking at the proteins available in a pure culture or from a patient sample. So this works by taking the patient sample, adding it to a metal plate, striking it with a laser that becomes ionized. The ionize are guided into this machine and it separates them. So kind of like electrophoresis, but on a kind of different scale. But it identifies them according to their mass to charge ratio. So how much ions they pick up based on how big or their mass, how much room they take up. So that mass to charge ratio is then detected as peaks, as we'll see here in a second. And it can also look at antibiotic susceptibility. So whether an antibiotic is able to treat a certain microbe. But we get something like this. So again, these peaks here indicate a high mass charge ratio. And so by analyzing them and comparing them, so in this case, this is a non-infected, so this is what a normal patient would look like. Here, if it's slightly infected versus highly infected, we start to see different peaks. So like, for example, with the highly infected, we have this area here that lights up that wasn't lit up so much in our non-infected patient. And then finally, imaging. So like MRI, CT, and PET scans are increasingly being used to look at deep tissue infections. So once the area is found, then it can be biopsied, and then this spares the patient from all of these invasive procedures that are kind of looking for them. So they can be identified before the procedure takes place.